let's jump on in. Cool. So let's see here. We left off our discussion of kind of the basic structures of how to science, the performance of research with time and research on Thursday. So that's a pretty short section. Hopefully we'll just run through this today and then kind of call it good. So time is, for every research study, a critical and inescapable piece of it, right? We're talking about, in explanatory research, these mechanisms, these sequences, these processes that take a bunch of time. Nothing functionally really happens instantaneously. So, when you're out getting water samples from recent hydrograph events and stuff like that, there's lag time. You don't need to be out there when it's raining to capture runoff, for example. You can go afterwards because there is a transmission time or a flow rate, both overland and subsurface flow. And when you are representing an understanding of a complex system like farm fields, like tile drainage systems and all of that, you have to take in time as a factor and recognize that the same study, exact same procedures, same equipment, maybe even the same technicians and stuff like that, done in 1990 versus in 2020, they would find different things even on the same sites with the same methods and so forth, because time has altered that site in some way. It's never going to be exactly the same. So let's talk about the effects of time in research and how to kind of work with or control effects of time. So most studies that we're going to be dealing with at the master's level, the most immediately important and interesting stuff, are going to be cross-sectional studies. And the idea of a cross-sectional study is that it is a single time slice. It is essentially a momentary snapshot. Now, a lot of cross-sectional studies happen over the course of a couple of weeks or a couple of months of field work, that kind of thing. It's very normal for a master's student, to sample in the spring and summer, and then come on back and analyze that stuff in the fall, write it up in the winter, graduate in the spring. That kind of a thing is a pretty normal setup because you're effectively, within a cross-sectional study, treating the span of time from, let's say, April 1st to September 1st. You're lumping the effect of time in all together and saying, okay, all of this happened summer of 2023, so we're not really treating time as yet another variable. And in the long run, even if you are paying some, you're splitting out the data between, this is the average rainfall for April, average rainfall for May, June, July, etc. The issue is that, 30, 40 years from now, maybe we don't care so much about what happened on day 1 versus day 131. We more just kind of care about what was the picture in 2023, and then 30 years later, climate has shifted maybe a little bit. And so what's the comparison year on year in that 30-year gap? So each one of those time slices is going to be a cross-section. If you think of a two-dimensional study where you have an input variable and an output variable, your x and your y, rainfall versus stream gauge level or something like that. And then imagine that Cartesian surface that you are plotting those findings in, and maybe finding some, positive correlation relationship where, x trends upwards, so y trends upward, and so forth. Time would be a third axis on that, and so you sort of mentally extrude the flat, infinitely thin surface of where you're plotting your results, and each time you remeasure, each one of those is another cross-section of the volume of data space that you're talking about. If you're thinking about data as a pipeline through time, each one of these sluice gates or check dams or slices or cross-sections is literally that. It is a cross-section across the volume that you are sampling through in time as time flows forward through that pipe. So cross-sectional studies, because it's a single slice, relative to other studies that, maybe you're remeasuring things 100 times in a row over the course of 3 months. That's a more complex study than going out one time and collecting a bunch of data one time and calling it good. Even more complex would be a longer-term study like some of these the ELTERs, long-term ecological research sites, where they are remeasuring stuff for a century. And they're remeasuring the same sites in the same ways to compare apples to apples and so forth, and moving things along in that way. Because sometimes you just need that long-term perspective, and you need the power to compare forward and backward in time instead of left to right or up and down slope. The variance is through time instead of in space. So cross-sectional studies, pretty quick. They're usually descriptive. Because an explanatory study needs an input to happen before, like the process starts and then modifies the phenomenon, creates whatever the outputs are. A cross-sectional study, because processes can't happen instantaneously, cross-sectional studies don't work well in that explanatory space, so they're usually more descriptive in that way. The opposite end of the opportunity space is the time series or longitudinal study. So time series can be as simple, so to speak, conceptually, as a repeated measures study. So a series of cross sections and just adding time in as a variable. That's pretty simple conceptually. Practically speaking, a flood will renovate part of the fields that you've been measuring for half a century, and now the measurements don't even make sense because part of it can't be used as a field anymore. So your sample size is shrinking over time in some cases. For time series or longitudinal studies of organisms, of trees, of people in a health panel study, something like that, people drop out, they move away, they die, they withdraw their consent from participating in the study for whatever reason. So conceptually, a repeated measures type study is fairly easy. You just redo the same thing, but practically speaking, those are really hard. And you can or cannot measure the same or track the same individual organisms. So in like a lab study with rats or mice or something like that, a longitudinal study over the course of the weeks or months of their lifespan, that's well controlled, and you get a lot of time slices, as many as your technicians can collect. But out in the real world, where maybe a farmer's family sells the field, that you've been taking measurements on, and the new owner doesn't consent to that, or they change the use of the field, maybe they plant something else that totally screws up the assumptions of your experiment, and it's just not useful to keep measuring that field. You figure out, okay, do we start measuring a different field? Do we just drop that one out and we keep measuring other fields? There are a lot of practical considerations that go into running a long-term study like that. And sometimes a master's student will parachute into one of these longer-term studies and do one time slice, and that's what they do their master's project on, and then they graduate, go on to do good things, and the next graduate student comes in and does the next time slice the next year, or whatever. That's a pretty common setup. Sometimes that's a very useful setup, especially for watershed type studies. So you may be involved in some previous research and plugged in with that, which is really nice in the sense that some of the development work, hammering out the bugs in your field protocol, figuring out where to do this stuff, figuring out what exactly to do, that's already been figured out when you parachute in and you're like the third or the fifth or the 500th time slice measurer. But that also kind of locks you into, you're obligated to continue measuring that in that way. So it can be good, it can be bad. It just kind of depends on how you're wired. If you like to be out on the bleeding edge of stuff, being in the middle or end of a time series type study is not particularly rewarding. If you're the person that is more concerned about not having to figure out everything for the first time and not being real sure of how to do that, then jumping in on a study like this is actually a really nice way to get started. So one particular species or subspecies of a longitudinal study is called a panel study. And the specific design here is that you track the same individuals with the same measures over time. This is a very powerful kind of study. There's a lot less variance, there's a lot less error in the data when you are tracking folks like this. So this is the kind of thing that big advanced governments do. A study will follow 10,000 people in a long-term Harvard study on marriages or whatever and see what happens over the course of 40 or 50 years, and you don't know how all those marriages are going to turn out until everybody's dead in those marriages, you know. So that's a very powerful and compelling kind of a thing. Cross-sectional data is usually broad, not deep. It's quantitative, not qualitative, more nomothetic versus ideographic. And in that way, with a single time slice of like survey information or a single field plot survey, you don't have super deep, you might have some detailed information, but it's not super deep. It's just very descriptive. A longer term health study, because you're remeasuring things, that can add some depth back into what is otherwise pretty superficial cross-sectional data, one-off data. So a lot of forestry studies are panel studies specifically because, you know, the same trees might be, you know, 400 or 500 years along in some old growth stands, and that's not a particularly big problem. Like, those trees aren't going anywhere, so you can easily remeasure the same trees and get really, really precise and accurate information out of them. The less rigorous but way easier to actually happen study is a cohort study, where you track members, you resample the larger group, but not necessarily remeasuring the same individuals. So that would be sending somebody into the same forest stand or the same county in southern Illinois and measuring the same area, but not automatically measuring the same trees, and probably some of the same trees, probably some of the same fields, but not exactly the same fields, not exactly the same trees, and so forth. So birth cohorts, if you've ever heard of generational studies that track, you know, differences between Zoomers and Millennials and Gen Xers and Boomers and silent generation folks and sort of draw a contrast between those are usually cohort studies, because you don't need to go back and ask the exact same people things. You can just ask anybody who's, you know, came up around the same age. You know, folks born the year that I was born can probably answer most of the questions in a similar way that I would answer them about anything, without having to spend the extra expense and risk of having to track me individually down, or you know, in failing to track me individually down, do we have to drop my data from the study because we can't re-identify me? So cohort studies, way more common. You have to generally go with a slightly larger or much larger sample size to control for the extra uncertainty, the extra error or variance in asking different people each time, or querying different fields, interrogating different trees in the same area of the same general species distribution, but not the same individual organisms. So that's fine, because you don't have to spend huge amounts of efforts tracking down the exact same organisms, individuals, whatever your unit of analysis is. You are freed up to go farther faster and collect bigger sample sizes typically, instead of lasering in on the same ones. So for a couple of examples out of my own research, at Acadia National Park, kind of the opposite direction. It's an island north and east of the mainland United States, whereas Hawaii is way south and west. one year we went up there and we spent all summer asking hikers about their experiences and observing their behaviors on a specific set of trail networks to figure out what was going on with those trail networks, where environmental impacts were happening, where people were getting lost or confused by visitor-created side trails, stuff like that. That was a one-off study. That was purely a cross-section. There was no intention, there's no need to go back and remeasure. Certainly not remeasure the same people, because like 99% of the people that we asked for the first study, the first cross-section, most of them are not going to be back the next summer. They're going to go to some other national park. So a longitudinal study was the wrong tool for that, when a cross-sectional study worked perfectly. We just needed a snapshot of what the problem was, how the problem was evolving, what was the pathology of the ecological impacts, the sort of the progression of disease of impact, if you want to think of it that way. And a cross-section is exactly right for that. That's a descriptive study. And parachute in, get the data intensively for 3 months, head out, analyze it, you're done. With Ozark National Scenic Riverways, just a little bit west of us, that was a longitudinal study. A professor that was here at SIU 30, 40 years ago, a guy named Ken Chillman, started to do a bunch of long-term monitoring studies at Ozark Scenic Riverways and capturing information about river use and stuff like that and how things were going. And about every 10 years, the Park Service would approach him to do that. He eventually retired, and they called me up in 2010 and said, hey, can you repeat this study again? That's a time series study of the cohort type, because again, it's different people each year that are there. And then the Ozark River User Studies, plural, so this study was one of those. All those considered together were a really sophisticated suite of cohort studies that let the Park Service defend some pretty controversial management decisions in court against some publicly organized advocacy groups that just disagreed with the government's management practices. So that's kind of how these different studies are used. Is it a snapshot problem? Can we solve the problem with descriptive information? Or do we need evolutionary changes over time, a longitudinal type series, a time series studies of a whole bunch of cross-sections? Or do we need to continuously track a panel of people and contact them kind of year-round every year? Do we need to continuously monitor and track how things are going with people as they circulate, or other organisms as they circulate into and out of a system? So if you're thinking about water, maybe liters of water as your unit of analysis in some field hydrology study, you're tracking different liters of water as they pass through the system, with different precip events and flow and stuff like that. You're not tracking the same water molecules each time. So that would be more of a cohort style study. All right. For the sense of sample size relative to time, that's another way to nuance or sophisticate your study. So a small n study is going to be that idiographic approach, where the time scale, it takes a lot of time, up to a couple of years of research and data collection continuously to do a good, thorough case study. But in that way, you're not treating time as a variable. It's a cross-section, but one that takes a couple of years, if you want to think of it that way, for a small n case study, a nomothetic study. For a large n study, where it's more descriptive or explanatory, the large sample size can be within each of those time slices, so contacting a ton of people, measuring a ton of trees, or that large sample size can come from lots and lots of time slices. So depending on whether you're doing a panel or a cohort, the sample size is really large, and also there are lots of samples. So it's the number of samples multiplied by the number of individuals in each of those samples. And this gets it can get pretty unwieldy to track individuals, and it's a little bit less of a pain in the butt with a cohort style study, but a longitudinal study, particularly a panel study, where you are dealing with hundreds of thousands to millions of data points, that is a lot of data management work. So in addition to the effort, the field sampling effort, there's a lot of effort that has to go into error checking data and clean and clear data custody. You always know who's got the authoritative set of the data, whether it's on your field laptop's hard drive or on a thumb drive on the way to the lab or in the lab computer. And then the lab computer is used to clean up the data, fill in gaps, drop incomplete data collection cases out of the data set, and that is now a different data set, having been cleaned and error checked and stuff like that, than the raw data set that exists on that field laptop. So it's very, very, very important especially in these large N studies where the data files get just ginormous, to have in your methods really clear, detailed, and well understood by everybody who's going to touch that data, who's doing what, where the authoritative copy of that data is at any given time, and what procedures you're going to do for error checking. What procedures are you going to do when part of the data file becomes corrupted and you can only salvage bits of it, and so on and so forth. So there's a lot of planning that goes into that side of things, even though it's not nearly as exciting or kind of sexy as going out there and conquering the world and collecting a bunch of field data. The legacy, the momentum, the inertia, the effort it takes to get a huge data set moving and collected and proofread makes that super, super, super important. So that's kind of the sense of time in research. Let's talk a little bit, let's contextualize this a little bit to your thesis so you kind of know which set of tools to pick up and use, and I'll kind of be it for today. So tell me about your project. Let's jump in.
So for N size, I'm not sure what it's going to be considered. I think it's on the smaller side.
Okay.
And I'm kind of doing the first slice of it.
Okay.
So it might end up being more of a cross-section type of study. But what I'm basically doing is I'm doing surveys of string morphology once a year, and then the difference in volume that we get for in between the years is going to be like the amount of erosion that occurred in this water year, I guess is what I call it. So I guess I'm collecting data for 2 years, but I only really have
one time slice to write your thesis on. Yeah, okay. So that's fine, right? I mean, that is what it is, and that's pretty much how most master's students get to deal with the variable of time. If you have 24 months and one internal summer season between the first 12 and the second 12 months, that's how it goes. That's how my research was as a master's student, and I had a blast doing it. No regrets. So that's totally fine. So yeah, you're getting the first cross-section in the time series, the first water year, and then collecting the second water year, but not necessarily having time to analyze it and add that into your thesis for comparison, contrast, and trending and stuff like that. It's more of a, hey, this is what we found the first year. This is my thesis. Yeah, okay, cool. So in some ways, that's a lot simpler to write. That's kind of nice. My wife is doing a bunch of writing up a bunch of parallel longitudinal studies with different mouse models in like concussion research, and it is just this crazy effort to just kind of keep everything straight. So sometimes it's nice to just have it a little bit more straightforward than that. There's nothing wrong with a cross-section study. This is how the longer thing starts, so it has to get rolling somehow. And for me personally, I'm wired that a lot of the interesting work is in the first wave. Repeated waves, less interesting. It's more of the same, There's not much new to figure out on wave 2, 3, and 4, and 5, and 10, but that's okay. So cool. What that means is going for like as large of a sample size as you can within the confines of this one sampling season that you're going to be writing about, and then collecting a second. What's your unit of analysis? Like, what are you running your statistics on? Is it each rain event? Is it each field? Is it each acre? It's not quite the same thing. So, what thing are you collecting data about?
I guess volume eroded per length of stream.
Okay.
That's kind of where it's at right now.
Okay, so you got a whole bunch of stream channels, and then chopping those up into like 100-meter sections or 1,000-meter sections or something like that?
I guess it's one channel, or I guess the main stem, and then we're taking 10 6-meter slices.
Okay, cool. Yeah. All right, so each of those is like something like a reach. Not really a reach exactly, but sort of like that. Okay. So your unit of analysis is a stream reach. And for each of those, you're going to get super detailed data with that cross-section. You're going to be able to describe what's going on with the shape, the volume of those spaces are in very, very high fidelity. And then with effort number 2, next season, go back and measure the same stuff in very high accuracy and precision, and then start to compare like year 0, so to speak, versus year 1, which is your second effort, to see what the change was.
Yeah.
Okay. Are you able to look at change within 1 year as well, or?
So, with the way it's being collected, I think I'm not going to have the number until the second year.
Okay.
Because the first year is the baseline.
Right. Exactly. Yes.
Sorry, did that answer?
Yeah. So that's pretty much it. So your unit of time is going to be year. In another study, where it makes sense, where something is more focused on like individual rain events, or with shorter-lived species like field mice instead of like marsh hares and stuff like that. The unit of analysis time-wise might not be a year, because you can have multiple generations of mice within a year, so it doesn't make sense. And if it were on a per rain event basis, then that's kind of a different setup than what you're doing with one cross-section per year per each of these 6-meter sections. Cool. So that's really important to kind of, as basic and obvious as that is, it's really important to have your sense of what is my unit of analysis? What's the thing? What's the phenomenon that I'm measuring? And how does that interact with time, or how do I think that interacts with time, so that I'm selecting the right window for how big my time slices or how thick my time slices are going to represent. In measuring like channel morphology, like stream riffles, for example, which are resculpted on a minute-by-minute basis in some sandy basins, a year-by-year time window makes 0 sense. But for something like this, where that volume change process might really be a little bit slower and might show up a lot better on a yearly basis, then you'd be essentially wasting huge amounts of effort to do thinner time slices. So as basic as sometimes this feels, it's so important to get after what's the appropriate time window that I'm looking for. What's the appropriate unit of analysis? Am I looking at 5 miles of river reach, or am I looking at 6 meters of stream channel? Cool, so that's it for now. We'll record this and pose these same questions to your colleagues remotely and see what they have to say about their studies. Otherwise, we'll see you on Thursday. Sound good? I'm around for questions if you have them, and we'll start looking ahead to continuing to write proposals and looking ahead to formatting proposals eventually into seminar presentations so you can practice those before getting them in front of your committees.